All right, everybody. Thank you, everybody that showed up. I guess it's such a nice, crisp morning. People stayed in bed. I don't blame them. Um, and I think at least I follow the weather a little bit. I think going toward the end of next week, I think we're supposed to get a little chilly, maybe even to the point of having a frost or two. We'll see. All right, but it's inevitable. All right. Apologize for the little issues on Tuesday. They got it straightened out. They had to reboot the system and change a couple of things. They got it working. Obviously, it's working. So don't forget to review the cancer lecture. And so we're moving on to our next system, which is the muscular system, right? Remember, when I was in the introductory slides and I said that, and when we were talking about membrane potential, and I said all living cells can maintain a membrane potential, but only two cell types have the ability to respond to that potential. The first one we talked about were neurons. Now we're going to be talking about muscle cells. All right. So we have muscles. What do they do? And there's different kinds of muscles, but in general, muscles have a number of basic functions. They enable us to move, which defines us as animals. So they're capable. They allow me to walk and talk, move my hands, and so forth. So they're capable and movement. They're also critical for you. They allow you to sit in your chairs, no matter what type of posture you use. There is the coordinated contraction, relaxation of various posture-related muscles. They are critical in stabilizing joints. For instance, here is my elbow joint. I've used this example before, so you're familiar with it. So we have our biceps. It has an origin and an insertion site, all muscles. So the biceps has its origin up here on my shoulder, which is closest to my main body, and its insertion site is here on my radius. It crosses this elbow joint. Hence, it tends to stabilize that joint, right? Lastly, as I've mentioned, we are homeotherms. We maintain a constant core body temperature despite variable environmental conditions. What in part enables this is the contraction and relaxation of skeletal muscles. This is, and one of the primary, I wouldn't call it a waste component, but a component of that process is the generation of heat, right? Now, again, the characteristics of muscles are they are excitable, which means they can act on that membrane potential. The next thing is they have the ability to shorten, so they can undergo contraction. They also correspondingly have the ability to extend beyond their resting length. So they're extensible. Think about that rubber band. And lastly, they are elastic. In not only are they extensible, but they will tend to return to their resting length. Now, there are three muscle types: skeletal muscles, cardiac muscles, smooth muscles. Skeletal muscles are by far the most numerous. Bulk of the muscles, most of the weight in your body is comprised of skeletal muscles. Biceps is an example, right? A cardiac muscle, we have one. That's the heart. I'm going to discuss in general the differences and similarities between these three, but eventually, for the rest, after the second slide beyond this, what I want to do is I want to focus on skeletal muscles. But that's not to say the cardiac muscle it's obviously important. You wouldn't survive without it. But I will focus on the cardiac muscle, the heart, when I get to the circulatory system, because that's a more appropriate place to discuss it. Similarly, when I talk about smooth muscles, they are usually associated primarily with tubular type structures like vasculature, gastrointestinal tract, and so on. I will get in more detail about those as I move into those systems. All right. So before I transition to, focusing on skeletal, let's talk about, what's distinctly different, what's the similarities, and so on. So basically, if we talk about skeletal and smooth muscle cells, they have similar structure in that they have, they're elongated, they're basic by bipolar, they stretch and have two ends. As opposed to cardiac muscles, which are branched. So there's there's similarities, there are differences. Also, the contractile aspects of all muscle cells consists of the interaction between actin and myosin molecules, which we'll talk about in a little bit. All right. Now, if we talk about terminology, I mean, you are obviously, your background is engineering, otherwise you wouldn't be in this class for the most part. So when we talk about jargon, every discipline has its unique jargon. Everybody does. Whether you come from, let's say, electrical engineering, mechanical engineering, chemical engineering, there are terms that you incorporate. And this is during your early study years. Biology is no different. So I haven't really focused on this to date, but I want to say that one aspect of biology that is helpful in acquiring this jargon is that there are prefixes and suffixes and so forth, which can facilitate your understanding. So in terms of terminology, if you see something with myo or mis in front of that word, it denotes muscle. Okay. So myoblast, blast means cell, myo means muscle, so muscle cell, myoblast. If you see sarco, it's the same thing. That means flesh, but it also denotes muscle. So for instance, you have a plasma membrane, correct? All cells have a plasma membrane. In the case of muscle cells, we refer to that as the sarcoplasma, which means the same thing, right? All of these things are referring to muscle. And you will see the use of this terminology going forward. All right, so basically, what are the differences? So skeletal muscles, they attach and cover the bony skeleton. For instance, here's our biceps. It's attached to my shoulder. Its origin in my shoulder. Its attachment point is on my radius, right? These muscles are striated. All you need to know right now, that means that you will see these light dark zones, and I will get into that a little bit further into this series. So don't worry about understanding what that is. Skeletal muscles are primary voluntary control. Obviously, I will my arm to bend, and therefore I can lift my forearms. It's under voluntary control. Now, the contractions, relaxation, so I'm capable of not using them at all voluntarily, or I can use them and I can, since they're overall controlling of body movement, they are capable of fairly rapid contraction relaxation cycles, but I have to slow down and rest at some point, and we'll get into that later. Cardiac, you have the heart. It continuously works. Otherwise, you're dead. Therefore, it is working while you are unconscious, i.e. asleep. So therefore, it is under involuntary control. Smooth muscles are similarly under involuntary control because they are controlling processes that you do not have voluntary control over. Those are your circulatory system, that is your gastrointestinal tract, and they tend to be associated with slow rhythmic contraction relaxation cycles. So they're not capable of fast responses, but slow responses. But considering what they're responsible for, that's very appropriate. All right. Now, both cardiac are striated as well as skeletal muscles. Smooth muscle cells, on the other hand, are not striated. Now, there are, for the next two slides, you will see this tabular formation where they're contrasting these three muscle types. Please go ahead and review it, though I am not specifically going to pull information from this to test you on the quiz or an exam. This is only for your own edification, all right? But basically, like I said, what I'm going to do, I want to focus this rest of this series of lectures on skeletal muscle, right? I'll grab the cardiac muscle. I'll grab discussions on the smooth muscle down the road, right? Now, so right now, what I want you to know going up for quizzes, going up for exams related to this lecture is this. Be aware of the three types of muscles. Be aware of the general differences, similarities that I talked about. That's it. Now, let's focus on skeletal muscles. All right. Skeletal muscles, they have 2 points of attachment, generally speaking. All right. So they have an origin, they have an insertion site. They cover body parts and they allow for general movement. Now, where do they attach? How do they attach? There are actually 2 ways in which they can attach, say, to bone. They can directly attach to bone. There is no intermediary. These tend to be rare. Most of muscles attaching to bone is via an intermediary structure, which we call a tendon. Tendons consist of primarily collagen type 1, but other collagens as well. And so the muscle then transitions to a tendon, and the tendon then transitions to bone. Now, these tendons, everyone's heard of them. You've heard of the Achilles tendon, right? That's your associated between the 3 muscles of my calf and my calcaneus, which is my heel bone. And that is, if you were to look at it, has a rope-like appearance. And actually, the term Achilles tendon actually consists of 3 separate tendons because there's 3 muscle groups. They come to, and they have each has their own tendon. They then are interbound together, and then they insert onto the calcaneus or your heel bone. All right. Now, so for instance, also the tendon that then inserts from my biceps into my radius is also this rope-like structure. However, not all muscle attachments are of this form. If we'll get into more detail, we'll get into the respiratory system, but the muscles between each of your ribs are called the intercostal muscles. These are think of those as we have these. Here's a rib, and the muscles are going like this. Now, they don't attach via these rope tendon structures. They have more of a sheet format because the nature of the muscle is different. All right. So you have the sheet type of tendons, you have the rope type of tendons. Tendons, again, they provide for attachment. They also they do not actively contract or relax. All of that is driven by the muscle. They have some elasticity to them as well, but they are actually creating these physical attachments, right? Now, what I want to do in this slide and subsequent slides is I want to discuss anatomy. We will start with the muscle organ, the tissue, which is remember, you have in physiology, you have tissue, you have cells, groups of cells, which we call tissues. If they're organized into a discrete tissue component, we call it an organ. Most of you, if I were to say what it suggests an organ, you would say the heart, the kidney, the liver, etc. Let me broaden that understanding. In physiology anatomy, every individual muscle, every individual bone are organs in themselves. Let me expand it further. Is blood a tissue? Is blood a tissue? Yes, it is. It contains cells. It is a liquid component, but it is still a tissue, right? Tissues don't think of this muscle, something like this that I can hold in my hand is a tissue. But blood, well, you can cup it in your hand a little bit, but blood is still a tissue, right? Now, let's talk about an individual muscle. Say the biceps, generally represented up at the top left here. And you can see on either end, there's a tendon. Those are attached to their respective bony attachments. And the muscle, note that the muscle consists of groups of muscle cells. And we'll get into what a muscle cell is in a moment. Right now, we're doing this gross anatomy of the whole organ. So there is a layer of tissue that then surrounds the complete muscle, and this is referred to as the epimysium. Within that, we have these sections, subsections of groups of muscle cells, and they're contained in their own matrix material, which is referred to, and each of those sections is called a fascicle. Within a fascicle, we have a group of muscle cells. Now, here is one end of muscle, here's the other end of the muscle, right? The thing to remember always is that a muscle cell goes from one end of the muscle to the next. It goes from here to here. It doesn't stop somewhere in the middle. Because remember, the purpose of skeletal muscles are contraction. So in order it can contract, that means that it needs to shorten. So that means you need to have a change in the length of that cell. It wouldn't do any good if that cell didn't go from here to here, but it stopped somewhere in the middle. There's nothing for it to pull on, right? So it goes from one end of the muscle to the next. All muscle cells go across the whole entire length, right? Now, next. So we have, and I'll go back and I'll show you a slide in about 4 slides. We'll get into more details here, but let's drop down for a moment. Because here's the next thing. The skeletal muscle cells are unique. I repeat, they're unique. When I gave, and during the introduction slide, I gave you a generalized cell, but I said this isn't all cells. So what's unique about skeletal muscle cells are a couple of things. Number one, they are derived from the fusion of multiple precursor muscle cells. Therefore, they have acquired everything that's in each of those constituent cells, including the nucleus. So mature muscle cells have multiple nuclei. Okay? Now, there is a structure that's unique to these skeletal muscle cells. They have a structure that is inside the cell called a myofibril. Do not confuse myofibril with a myofiber. Myofiber is another word for muscle cell. Myofibril is a series of the smallest contractile units, the sarcomeres, and they also go from one end of the muscle cell to the other end of the muscle cell, right? So the actual skeletal muscle cell is filled with these myofibrils. Each of these has contractile properties. And as we increase the number of these myofibrils, we obviously increase the contractile force within that muscle cell. All right? We'll get into more details later. Right now, I'm just talking about some anatomy, introducing jargon, and so on. Okay? Now, these myofibrils is what contracts. And since they're in the muscle, as they shorten, that means the overall muscle will shorten. Now, and within a muscle cell, when we induce the contraction of an individual myofibril, all the myofibrils within that muscle cell will contract. You don't have one contracting and all the others not. And we'll talk about cell signaling later. But right now, skeletal muscle cells are filled with these myofibrils. All right? Now, I mentioned the smallest contractile unit, the sarcomere. That's what you're looking at right here. So the sarcomere, you have a series of those sarcomeres which comprise together an individual myofibril. Okay? So the sarcomere starts at what is called a Z disc, and they end with a Z disc. 2 adjacent sarcomeres share the same Z disc. So we have sarcomere, 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 sarcomere. Okay? And we have these than these Z discs as well. So each of these sarcomeres within a myofibril will contract basically at the same time. That means it shortens. Okay. Now, so let's talk just a little bit about the anatomy. We'll worry about functionality in the next few slides. So Z disc to Z disc, right? What is attached to that Z disc? If we look here, this structure here is called the thick filament, and it's comprised of myosin molecules, right? It's anchored to the corresponding Z disc via elastomeric elements that look, those are those little yellow springs you see, right? Now, next we see these little blue chains here. These are the thin filaments, which consist, among other things, they contain actin molecules. And they are rigidly attached to the Z disc here and here. But they don't, under relaxation, there is a gap here because they have a fixed length, right? That's all I want to talk about at this particular moment, all right? Except to say that the thick filaments contain molecules of myosin. These are heads of the myosin molecules, and they are designed to grab a hold of actin molecules, undergo a power stroke, and if it's hard to see here, but the heads are pointing, so the heads to the right are pointing to the Z disc over here, the heads to the left are pointing to the Z disc over here. When they undergo a power stroke, here is either end here, they move like this. So that means they're grabbing hold of these actin filaments and pulling them toward the middle. All right. And that is going to result in contraction or a shortening of that sarcomere. Even though these sarcomeres are very small. They're very small. Let's say that the contractile shortening capacity of that is on the order of microns. However, an individual myofibril can consist of hundreds or thousands of these sarcomeres. So even though the contractile length, shortening length, is very small, it scales because we multiply it by the number of sarcomeres in that series. So that results in this. Even though they're very, very small gaps that they can contract to, if we talk about my biceps, okay, remember the muscle cell goes from here to here, so therefore the myofibril goes from here to here. When it shortens, that muscle difference changes enough to lift this. So it can actually shorten by centimeters because of the scalability of that series of sarcomeres, right? So let's go back and just do some general anatomy. Here again is a muscle organ. There's over 200 muscles in your body, skeletal muscles in your body. Now, again, it's attached to bone via a tendon. Here is the membrane that defines this muscle is called the epimysium. Here are individual fascicles, and they are defined by a membrane called a perimysium. And individually within the muscles, here's the muscle cells. The muscle cells are separated by an endomysium that's between each of the cells. And if you remember when we were talking about a nerve, it looks somewhat similar, right? You had neurons that had their own individual fibrous material encapsulating them. And what did I tell you? The reason why is I told you it acts as an insulator. Same thing here. Because remember, electrical communication stimulation of both these. Right? So that's why they're isolated. Because you don't necessarily, and as we'll get toward the end, we don't necessarily, you can stimulate any number of cells. You don't have to stimulate all the cells at the same time. Your body has the ability to stimulate some and then gradually scale up to more and more. All right? And we'll talk about why later. But I will say this. Within a cell, all of the myofibrils will contract at the same time. but not all cells have to contract at the same time. Depends on how they're controlled, which we'll get to later in this series. The next thing I want you to notice is this. It's not showing that, well, yeah, it is to a certain extent. I told you in the introductories that all, with the exception of cartilage cells, all cells in the body are within basically 50 microns of a blood supply. All right. So if you have a tissue that's larger than a millimeter cubed, it's got to have a vascular supply. Your biceps muscle is obviously much larger than that. So if you also look at the anatomy of a muscle, just like when you were looking at the anatomy of a nerve, it's big, it's got a vascular supply. We also have a nerve supply because that's what's going to control contraction, relaxation, right? So it's got all of that. Okay, let's focus on that muscle cell itself. I already told you that that muscle cell is filled with myofibrils. Let's talk about something else a little bit more different, where it's more complex than that. It has its own plumbing system. All right. So if you look here, the surface of the sarcolemma, the plasma membrane, you'll see little holes. Those holes are open to the interstitial fluid outside the cell, right? And they are internalized into something called a transverse tubule, because that tubule transverses into the cell, all right? And it's open to the outside of the cell. And these openings are basically, the T-tubules are associated over the Z discs of each of the sarcomeres, all right? So we have these holes, and they allow for, so that's what you see in the purple here, all right? And that's part of the transverse tubules. And basically, so this is a Z disc. This is setting over a Z disc. So the purple is the transverse tubule, or the T-tubule. And then on either side, see these blue network structures? This is a different structure called a sarcoplasmic reticulum. Okay? So we don't worry about function right now. This is again just anatomy. So this blue structure is called the sarcoplasmic reticulum. This purple structure is called the T-tubule, or the transverse tubules. Now, they are not physically attached, but they're closely associated. We'll talk about control later. Bless you. So basically, the T-tubule, its purpose is that since it is exposed to the outside surface, it actually represents an internalization of the plasma membrane to deep inside the cell. We'll talk about why we need that later. Again, I'm just focusing on the anatomy right now. All right? So we're going to be revisiting this in a bit, but what I want you to realize is this. Within a muscle cell, we have these myofibrils. Myofibrils are a series of connected sarcomeres, the smallest contractile unit. And associated with the Z disc of every sarcomere is this internal plumbing system. The T-tubules are circular, surrounding that, and then we have the sarcoplasmic reticulum. All right. Let's go back just a moment. Why multinucleated cells? What are skeletal muscles multinucleated? Two reasons. The first one is that, as I've already mentioned, we have multiple precursor muscle cells fusing together to make a mature muscle cell. Why? Why? Why not just have a whole bunch of little bitty muscle cells? It's all about efficiency of regulation and control. It's much easier to control one big cell than 100 little cells. All right. So basically, it allows for scale. So it just makes it easier to control. Instead of having each cell having its own myofibril, we have these larger cells with multiple myofibrils, and they're all contract. Each cell contracts all of 100% of all the contractile units. So sarcomeres inside contract at the same time. The more I have, the more force I can generate. Okay, so that's why we have these multinucleated cells. These are not the only multinucleated cells in your body. There are others, but this is the first example that I'm covering. All right. Now, let's zero in and talk about the interrelationship of the sarcomere, because you have to understand its anatomy, how it changes, because if you don't understand that, you can't understand how your muscles, skeletal muscles work. All right. So we go back up here. Again, a slight revisit, but I'm sorry, I don't think that repetition is a waste. I think it helps to reinforce stuff. So I will be repeating myself. All right, so here is a muscle cell at the top. Note that we have the sarcolemma, or the plasma membrane. We have the nuclei. Notice that they are right underneath the surface of the cell. That's because they're excluded and squeezed out because of that cell is filled with myofibrils. Right? Not only, and so first off, here is where striations come from. I mentioned that skeletal muscle cells are striated. They have zones of light, dark, light, dark, light, dark. The dark is associated with the thick filaments. Those are the myosin filaments. The light zones are where they're not. That's basically it. So the dark red is where you have a thick filament. The light is where you don't. Right? One is the presence, the other is the absence of myosin, or thick filaments. Now, if we take this myofibril and extend it out, it is nothing but a series of sarcomeres. Hopefully I've drummed that into your head, because that's absolutely critical. All right. And remember that it's a whole series of sarcomeres where a Z disc is shared between two adjacent sarcomeres. Right. So sarcomere, 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 Z disc, Z disc, Z disc, Z disc. Now let's talk about a single sarcomere. A single sarcomere is Z disc to Z disc. Okay. Now, so basically, what I want you to do here is Z disc to Z disc. Right. Now, these terms commit to memory. Commit to memory, because you will be seeing them multiple times. All right. So we have Z disc to Z disc. So this is an entire sarcomere. Right here in the middle, we call that the M line. M for middle line. All right. M line. Really hard to remember. Right. So this is the M line here. Right. Now, the thick filament, that dark red, represents the myosin molecules all grouped together. Okay. Now, they are anchored to respective Z discs via an elastomeric element. We'll talk about that in a moment. But basically, they're held in place at the M line. They don't move, basically, within the sarcomere. They are fixed within the sarcomere. Next are the thin filaments, and they are physically attached to respective Z discs, but they're open. They're not attached to anything toward the M line. Now, what happens during contraction is the myosin heads grab a hold of the actin, undergo a power stroke, and pull the respective actin molecules toward the M line. And that results in a shortening of the sarcomere. Right? Now, we have something called the A band, which basically represents the myosin molecules. This is fixed. They never change. They don't change in length. So the A band never changes during contraction. This is under relaxation at this present time. Now, we have something called the I band, and the I band straddles the Z disc. Remember, we have a sarcomere here, sharing a Z disc. The light band is that zone which is not occupied by a thick filament, right? The middle is a Z disc, and then we have a Z disc, an elastomeric air element, and then a thick filament on either side. Now, during contraction, what can happen is that this I band can get smaller because we're going this way. As this doesn't change, this moves closer, this can get smaller, but can never go away because the Z disc persists. Right? Now, what about the A band? The A band is defined as that area not occupied by the thin filaments. Okay? During relaxation, there is separation here. So that represents the A band. This area here represents the A band. During contraction, they can actually overlap, so it can actually go away. All right? So I'll keep revisiting this, but you might as well commit this part to memory, right? Because this is really important, right? Because this is the basis of contraction and as well as relaxation. Now, again, we come down here. Here's a cartoon, a simplified version of a sarcomere, Z disc to Z disc, thick filaments attached physically via these elastomeric elements. That actually allows for elasticity. That allows the sarcomere to stretch beyond its resting length. And it also gives this rebounding like in a rubber band because the spring element wants to return back to its resting length. All right. The thin filaments, which contain actin, are physically rigidly attached to their respective Z discs. But they're floating over here. They're not bound to anything here. All right, so the sarcomere. Now, let's look at the actual thick and thin filaments. What are they? I told you one is basically myosin, one is actin, but it's a little bit more complex than that. In the case of the thick filaments, they are bundles and bundles of individual myosin molecules. A myosin molecule consists of an interwound set, two molecules of a head and a tail. The tails are interwoven. These tails then are integrated into bundles and exposing the heads along the surface of the thick filament. Again, the role of the head, myosin head, is to bind to actin. Now, let's talk about the thin filament. It's a bit more complex. Again, obviously, these little blue balls are the actual actin molecules. Think of those as little globules, little spheroids, if you will, just for the sake of simplicity. They have to be held in place. They're held in place in the thin filament by something called tropomyosin. It's protein tropomyosin. So that binds to the actin molecules and holds them in place. The next thing you see these little group of yellow balls, which is called the troponin complex. This plays a critical role in initiating contraction and relaxation. We'll get into that in a moment. Again, I'm just focusing on the anatomy. So again, thick filament, bundles and bundles of individual myosin molecules. And so these bundles of the tails are all bundled together and the heads are all exposed out. All right. So the next thing is we have an M line here, which is the middle of the thick filament. And then on either side, the heads tend to point to the respective Z discs. Right. The thin filament consists of actin, which are held in place by tropomyosin strands. And we have something called the troponin complex, which is involved in control. Okay, let's put it all together. I've talked about them. Now let's just talk again. I'm just reinforcing. So here's a muscle cell cutaway. Here is a myofibril. What I want you to note here is the hole that is the entryway of the T-tubule. Then that internalizes this out. So this is inside the lumen is actually part of, it's actually communicating directly with the interstitial fluid aspect. All right. So we have our T-tubule. And our transverse tubules go all the way through the cell and they surround each of these myofibrils. This blue structure in between is the sarcoplasmic reticulum. Right? Now, what you have here is these are bundles and bundles consisting of all these sarcomeres. Right? So let's go back up here. We have our A band. The A band is the thick filament length that does not change. We have the I band, that is the area which is not occupied by a thick filament that is bridged across the Z disc. We have the H zone, which is that area around the midline, which is not occupied by the thin filaments. Let's see. I think that pretty much covers that. Now, let's put all this anatomy together and talk about function. How does it work? All right. So, muscle contraction and relaxation. This is the ability of the muscle cell to shorten and then return to its resting length. So it's at a resting length. It is induced to contract. So that means it shortens and then it can relax again. All right. Now, this process, if we thought, it's under nervous control. It's voluntary nervous control. So that means there is a signal coming from the nervous system to initiate contraction. Right? And which means a physical shortening of the sarcomeres. So what I want to do is let's talk about what that process is and how you return back to a resting length. Right? So what we refer to this is the sliding filament model of contraction. Again, the sliding filament model of contraction. As I told you when I was talking about the anatomy, the goal of the myosin heads is to grab a hold of the actin, undergo a power stroke, and pull the actin toward the M line. Since the resting length of the sarcomere is here, as I pull the actin toward the M line, the actin is permanently bound to the Z disc. That means the Z disc gets closer together, which means that distance shortens. Since they are a series of sarcomeres that scales, that results in an overall shortening of that muscle cell. Since that muscle cell is attached at either end of the muscle organ itself, that means the overall muscle organ shortens. Right? Now, so let's look up here. At the top, we have a transmission electron micrograph, and then down below is a defining cartoon anatomy slide. So this is at rest at the top. So our Z discs are here. Okay? The myosin is not binding or holding on to the actin. So we're under relaxation. When we stimulate activation, contraction, we'll talk about that later, how it works later. What's going to happen is that, let's say that here's the M line here. These are the heads of the myosin pointing toward the respective Z discs. And under contraction, they're going to grab a hold of the actin, undergo a power stroke, pulling the respective actin molecules toward the M line. All right? What's happening is that this repeated contraction means that the actin molecules, the thin filament, is being pulled along the surface of the thick filament. And that's where the sliding part comes in. So it's sliding across the top of that thick filament, right, toward the M line. So what happens is, so if we look up here, note that we have the A band is from here to here. The I band is from here to here. The H zone is from here to here. All right. So during contraction, what changes? You will have a question on this, so you better know it. So during contraction, what happens within the sarcomere? Okay. Number one, the length of your thick filament, which is your myosin, does not change. It never changes for all intents and purposes. So your A band doesn't change in length. What happens over here in your I band Now, because this doesn't move, but these thin filaments move toward the M line, right? As they're moving toward the M line, they're going to be pulling the respective Z disc toward the M line. What that means is the distance between the thick filament on two adjacent sarcomeres is going to get closer together. So that means the I band gets shorter. But there's a limit, because the Z disc and those elastomeric elements never go away. So you can never go to nothing. You can get smaller, but there is a limit to how small you can get. All right, that's the I band. The H zone is the distance within around the midline where you do not have the presence of a thin filament. So under relaxation, there's a gap there. But as you're moving the respective thin filaments toward the M line, what happens? They're moving toward the M line like this. What happens is that they can continue to move until they are actually overlapping. What this means is that the H zone not only can get smaller, it can actually go away. All right? So you need to be aware of what changes, what doesn't change. The A band does not change in length under contraction. The I band gets smaller but doesn't go away. The H zone gets smaller and can go away. All right? And this is all part of the sliding filament model of contraction. This is how a sarcomere basically works. And because this is scaled, remember, this is a single sarcomere within this entire length of a myofibril. You had a whole series of sarcomeres. So this all scales. So even though this sarcomere can only contract this little bit of length, it scales for every single sarcomere that's in the length of that. So this can actually translate to centimeters of shortening of the overall muscle organ itself. Okay? Now, this is what I just was talking about. I'm showing it to you because I expect you to know it. So use whatever, all the stuff I've given you, understand and know it. If you do not understand how the sliding model works, you can't understand how a muscle works. Right? Okay? Now, how do you start? How do you initiate a contraction? So your muscle, your skeletal muscles their resting state is relaxation. Their resting state is relaxation. To initiate contraction, the myosin has to grab a hold of an actin. So under relaxation, this process is inhibited. So the default is the myosin wants to grab a hold of here's an actin molecule, here's the head of a myosin. It wants to grab a hold of that actin, undergo a power stroke. It can't. Why can't it? Because there's something in the way. There's something blocking its access. So even though the myosin wants to bind to the actin, it can't gain access. This is what the troponin complex is there for. Okay? So I don't want to go into too much detail, just enough for you to understand. All right, so this is instead of looking along the length of the sarcomere, we're looking at it face on. All right, so we're doing a cross section. So here is basically, if what you're talking about here's the myosin heads here on each side. These group of balls here, these three groups of balls is the troponin complex. The orange is the tropomyosin. Remember, that's a continuous strand of which all the actin molecules are bound to. Think about a pearl necklace, all right? Now, under relaxation, the myosin head cannot access its binding site on the actin molecule because the troponin complex is in the way. It's physically in the way. It's physically blocking the binding of myosin to the actin. So therefore, since I can't bind myosin to actin, I can't undergo a power stroke, I cannot contract. To initiate contraction, I got to move the troponin complex out of the way. How does that work? All right. So if you look at this troponin complex, see these little red balls on the surface here? Those are actually calcium molecules. So under relaxation, I repeat, under relaxation, there are calcium binding sites on the troponin complex. They are not saturated. They are not saturated. Okay? So to induce contraction, I need to saturate that. So that means I have to increase the calcium level in the cytoplasm of the muscle cell. The increased calcium binds to its receptors. When they are saturated, as we see here, it actually results in a conformational change of the troponin complex. It physically moves out of the way, exposing the actin binding site. The myosin grabs it, undergoes a power stroke. We have contraction. Now, as long as those calcium molecules are occupying the binding sites, we will have contraction. This is where things start to deviate from nervous system communication. So, as long as that calcium is there and binding to the sites, we will have contraction. In order to induce relaxation, I have to get rid of the calcium. So that's where we're going to go next. Okay. So we know what a sarcomere is. You know the parts of a sarcomere. You know what moves, what doesn't move. Now we're getting in, it's controlled by calcium. Calcium results in a conformational change in the troponin complex. It now exposes, it moves out of the way, exposing the myosin binding site on the actin molecule. Myosin binds it, undergoes contraction. But now, in order to do relaxation, we got to remove that calcium or reduce its concentration. Right? But in any case, what you need to do is, before I get to that control mechanism, remember, here is my forearm, here is a myosin head. My fist is an actin molecule. So what happens, this is cocked. When it grabs an actin molecule, it undergoes a power stroke, moving toward the M line. So it's grabbing this and moving toward the M line, right? Now, since we're talking about these physical head movements of these molecules, this requires energy. This requires ATP. So what happens is the aspect of cocking and then recocking involves the expenditure of energy, ATP. So to contract and relax, it requires the expenditure of energy in order to prepare for the power stroke and enable another one. All right? I don't want to go into details. I just want you to be aware of that. All right? Now, how do we control contraction relaxation? Obviously, muscles are excitable cells. What controls them is going to be the nervous system. So we have an upstream neuron, an afferent neuron, which innervates a muscle cell. All right? With me so far? So we have an action potential coming down the axon of that presynaptic neuron. When it gets to the terminal axonal bud, what happens is we have an action potential coming down the length of the axon. We get to the terminal bud. What we have there, we transition from voltage-gated channels from sodium to voltage-gated channels for calcium. Calcium flows into the terminal bud. This initiates exocytosis and the release of neurotransmitters into the synapse. These neurotransmitters diffuse across the synaptic cleft. They bind to receptors on the postsynaptic cleft. But remember now, the postsynaptic cleft is a muscle cell, not a neuron. And it will initiate a chemical-gated channels. In this case, say sodium flows in, and the sodium will initiate a graded, a different response on the muscle cell. All right? This is where I got to when we're in the nervous system. I said at the level of the synapse, There is no difference in neuron communication with either a neuron or a muscle cell. Now you're going to find out after this point, it's going to get a lot different. It's going to get a lot different. Now, the things that you have to remember when we were talking about neuron potentials, what we're talking about is the exchange of charge particles across the surface of the cell. All right. So when we're talking about electrical communication in regards to a nerve cell, it's all about surface changes in charge. When we're talking about muscle cells, that's different because we have to stimulate all of the sarcomeres. Where are the sarcomeres? Deep inside the cell. So we have to get an electrical signal which is restricted to the surface of a cell deep into the cell. How do we do that? That's where the T-tubule comes in. So it allows us to internalize that surface phenomena because the T-tubule actually represents a continuation of the plasma membrane deep into the cell. So those membrane-associated electrical events get internalized and have the capacity to stimulate every sarcomere within the cell. All right? So it all begins the same as for neurons, but it gets a little bit different, right? So how does it get different? It's this way. We have our presynaptic neuron having an active potential, translates to neurotransmitter release in the synaptic cleft. We initiate a corresponding active potential on this muscle cell. It stays on the surface of the cell, of the sarcolemma. It's going until it encounters a T-tubule. Since the T-tubule is basically a plasma membrane, except it goes transverse, it gets internalized. And it goes all the way in. Remember, they're setting over, these two tubules set over every sarcomere. As it gets down to that, it communicates with the sarcoplasmic reticulum. So we have this potential coming down the T-tubule. As it encounters the sarcoplasmic reticulum, this electrical signal and a voltage-gated signal will result in an opening of calcium gates in the sarcoplasmic reticulum. The concentration of calcium is greater in the sarcoplasmic reticulum versus the cytosol. So that means when these open, calcium flows out of the sarcoplasmic reticulum into the cytoplasm, increasing the cytoplasmic concentration of calcium. That means that the calcium saturates its binding sites on the troponin complex. The troponin complex undergoes a change such that it now exposes the actin binding sites on the myosin binding site on the actin molecule. Myosin binds, undergoes contraction. All right? But if you remember, I emphasized that we have to have relaxation. What is relaxation? The way you reestablish relaxation is you've got to remove the calcium from the cytoplasm so that it will be released from its binding sites on the troponin complex. And then the troponin complex will move back and disengage the myosin from the actin. Now, what does this comprise? How do you do that? So what you have to do is you have to move the calcium from the cytoplasm back to the sarcoplasmic reticulum. Wait a minute. I told you that the gradient is from the sarcoplasmic reticulum to the cytoplasm. If I want to move it the other way, the only way I can do that is by active transport. So I have to actively transport the calcium from the cytoplasm to put it back into the sarcoplasmic reticulum, right? This also takes time. It also takes energy, additional ATP. So, until the calcium levels in the cytoplasm drop low enough so that the calcium can dissociate from its binding site on the troponin complex, I will have contraction. When I do reduce it by pumping it back in the sarcoplasmic reticulum, then and only then can I have relaxation. So once the calcium levels are reduced, the binding sites are not saturated, the troponin complex moves back to insert itself between the actin and the myosin, and it disengages the myosin head from the actin, and we have relaxation, right? Translate that to change in length of your sarcomere. Everybody with me so far? Okay. So I like to use the example of rigor mortis to try to drive home the message. Everybody, I mean, you like to watch murder shows, you know, but no, okay. But no hunters here in this audience, probably not. But anyway, let's say, so basically, let's say that you are a detective. Sherlock Holmes, favorite detective. Doesn't matter. All right. You're coroner, whatever. So basically, what happens is this. You are called, there's a dead body. You go to the dead body. One of the first things you have to do is assess time of death, estimate time of death. Okay. Now, how do you do that? Normally, what you do, there is, you have a thermometer, you stick it into the corpse, and there's a sets of equations that, depending on the size of the individual, the temperature the temperature difference between on deep inside the person, the corpse, and out here, you can estimate time of death. All right, because you have this massive tissue, it's losing heat, and it's a function of surface to mass ratio with the corresponding temperature difference out here. The colder, the greater the difference, the faster you're going to lose temperature. But there's equations where you can plug all these temperatures in, and it'll tell you approximate time of death. Oh, you forgot your thermometer. Okay. Oh, screw it. Your assistant forgot to put it in the van when you left. So you can still approximate time of death because of the process of rigor mortis. All right. So what is rigor mortis? Rigor mortis, this is based on, I told you that what happens once you initiate contraction, in order to reverse that to relaxation, we have to get rid of the calcium. All right. As long as the calcium is there, you will have contraction. So how does that relate to rigor mortis? You have your person is killed. Now you have a corpse. Okay? It's dead. So that person or animal, whatever it is dead. Now what happens is that at the point of death, things start to change. However, things change at different rates. One thing that degrades first or early on is the membranes that comprise the sarcoplasmic reticulum within the muscle cells. Remember what the sarcoplasmic reticulum is. It is a reservoir of calcium. All right, the calcium is stuck in there, so that means that we don't have relaxation. But shortly after death, hours after death, what happens is these sarcoplasmic reticulums degrade, so they're no longer functional. They can't confine the calcium. The calcium then leaks out, increases the calcium in the cytoplasm. It binds to the troponin complex. You don't have to be alive for this. Everything's ready and cocked and ready to go. It doesn't matter that you're dead, but you're dead. The calcium then saturates the troponin complex, and we have contraction. Now, we cannot have relaxation because of two things. Number one, you no longer have a functional sarcoplasmic reticulum. So it's always, all those holes are permanently there. Oh, even if it wasn't, you're dead. There's no more ATP, so I can't pump it back in, even if I could. even if I had a working sarcoplasmic reticulum. How does this translate to rigor mortis? So in rigor mortis, what happens is, okay, the murderer killed you and stuck you in a trunk. You know what a trunk is, a confined space. In order to do that, he had to kind of crunch you up like this and put you in that trunk. So when they took you out of that trunk, you were like this. Okay, no matter, you rolled around, you were like this. It was really difficult to kind of unbend your arms. That's rigor mortis. This happens within hours of death because of the degradation of the sarcoplasmic reticulum membrane, the release of calcium, the induction of contraction, but we can't relax. Now, what happens hours later, many hours later, is that, remember, the first thing to degrade was the sarcoplasmic reticulum. The second thing to degrade much later is the sarcoplasmic, the plasma membrane of the muscle cell. All right? As it degrades and opens up, gets big holes in it, the calcium leaves the cell. As the calcium leaves the cytoplasm and drains out, that means that the troponin complex, the calcium is removed, and we have dissociation of the actin myosin, and we have relaxation. So, when they dump the corpse out of the trunk, is it like this or is it like this? If it's like this, the time of death, it all depends on the temperature. You can approximate it some hours after death. If you're a limp thing, and that means that it has many hours later or even days later, right? There's other things in addition, but this is not a forensic show. So we won't go into the more details on that. Just focusing on rigor mortis is to drive home the message that we have to physically remove the calcium from the cytoplasm in order to have relaxation, right? Okay. Since we're talking rigor mortis, since we're talking murder, let's talk about poisons. All right. So let's talk about neuromuscular toxins and poisons. How do they work? All right. So what they do is they are designed to disrupt this communication cascade that can lead to either contraction or relaxation. All right. So pesticides, insecticides, what we use to kill insects with. All right. How do they work? What they do is they are designed to disrupt neural to muscle cell communication. So remember, it's all about the synapse. We have our presynaptic neuron commuting to our postsynaptic muscle cell. Remember that within that synapse, what do we have? Within that synapse, not only do we have the receptors that bind to the neurotransmitter on the postsynaptic membrane, we also have this large amount of enzymes which rapidly degrade those neurotransmitters. Remember when I was talking about the nervous system and I said what happens, you have an action potential, it induces the release of a neurotransmitter into the synapse, but it rapidly degrades. So it only can give you a little bit of signaling before it's gone. In order to signal it again, there needs to be another action potential. And remember we talked about action potential frequency. All right? Now, in terms of muscle contraction, we have our presynaptic neuron having an action potential coming down to the synapse. It induces the release of neurotransmitter. We have a moment of contraction because that gives a signal. Sarcoplasmic reticulum opens. We get contraction, as we've already talked about. Then there's relaxation. In order to continue the next level, I have to have another action potential coming down. All right? What happens if I target those neurotransmitters? What happens if I add a poison that actually blocks the degradation capacity of those enzymes? That means that those neurotransmitters don't go away. That means they always bind to their chemical receptors and are stimulating contraction. So that means that when they're in place, even though I've only sent a single signal, I cannot have contraction relaxation. Right? So how does that work? An insect is no different from you. It breathes air. And breathing air means that it has the equivalent of lungs for you that have the ability to pull in air and dispel air. In other words, you have inspiration and expiration. One requires contraction, one requires relaxation. Now, in this case, with the way these pesticides work, they induce a constant state of contraction. Think about that rigor mortis. But that means you cannot bring air in and expel air. So you can have gas exchange. So the way pesticides work is by suffocation. You can't breathe. Okay. And they can work on humans too at a large enough dosage. But in any case, all right. Next, tetanus. Everyone here probably has had most of you have had a tetanus vaccination. All right. If you do anything in medicine, you will have a tetanus vaccination. And I am old enough. I remember when tetanus vaccinations you had to have every year, every 2 years. It's every 10 years now, soon to be a lifetime. Now, why is that important? It's because bacteria, Clostridium bacteria, is extremely common in the environment, especially in the dirt. All right. And if you get inoculated by this Clostridium bacteria, it will release agents that will block the release of glycine, which actually is critically important for controlling relaxation. So what that means is this will induce contraction and inhibit relaxation. And tetanus is also referred as lockjaw because classically it would be associated with the mandible, such that you would have contraction and your mouth would be constantly shut and you couldn't eat. You would actually starve to death, right? Unless you bust a hole in the teeth and stick a feeding tube in. But tetanus so that's where tetanus comes in. That's how it similarly to these pesticides, but by different mechanisms, it will induce contraction and inhibiting relaxation. Then we have the opposite. So what we can talk about are poisons, which will, such as curare, which will basically block the ability. So basically what it does, it binds to the receptor. So here's the synapse. Here's the presynaptic, postsynaptic membrane. Here are the neurotransmitters. They bind to the receptors on the postsynaptic membrane, right? What curare does, it binds to those receptors. It blocks the binding of the neurotransmitters, but it does not transduce a signal. So in other words, those chemical gates don't open, which means that you can't get stimulation of contraction, which means that you are in a constant state of relaxation. In either case, whether you are in constant contraction and can't relax or constant relaxation and can't contract, you still die due to suffocation because you can't have gas exchange. All right? All right, I'll go through this and then I'll quit because we have seminar in a little bit. So during this process, when we were in the endocrine portion and I talked about regulation of plasma calcium levels, I said that it's important. They're critically important for multiple physiological processes. What are those processes? Number one, they are critical for neuron to neuron or neuron to muscle cell communication because the calcium influx into the terminal bud of the axon initiates exocytosis and the release of neurotransmitters. Number two, calcium plays a critical role in contraction. because it has to saturate its binding sites on the troponin complex in order to permit contraction. Right? There are other reasons, but those are the two primary ones I want you to be aware of because they play a critical role here for skeletal muscle cell communication. All right. With that, I'm going to stop because where I want to go is going to take more than 5 minutes, and I don't want you to be late for seminar, although it's just in the same building and not that far away. All right. So that's it for today, and we will convene again next Tuesday.